Hello everyone, welcome to Cyber Slate. Our today's topic of discussion is osteoarthritis. So let's get started. Now, pathophysiology. But before this, I will like to tell you about lesions. So what are lesions? Lesions forms from degeneration of articular cartilage and its disordered repair. Now chondrocytes. So what are chondrocytes? Chondrocytes are the cell which has secreted the matrix of cartilage and becomes embedded in it. And it produces type 2 collagen and proteoglycan. The type 2 collagen is basis for articular cartilage and hyaline cartilage. And proteoglycan are proteins and major component of extracellular matrix. So what are their functions? Type 2 collagen provides resistance to tension, and in other hand, proteoglycan provide resistance to compression. In osteoarthritis, pathological changes occurs in chondrocyte and matrix. So there are three phases of chondrocyte changes. First is chondrocyte injury, which is related to genetic and biological factors. Second is early osteoarthritis, and third is late osteoarthritis. Now, in early osteoarthritis, chondrocytes proliferate and secrete inflammatory mediators, collagens, proteoglycan, and protease, which act together to remodel cartilaginous matrix and initiate secondary inflammatory changes in synovium and subchondral bone. Now, what happens in late osteoarthritis? Firstly, there is repetitive injury and chronic inflammation in the synovial joint, which leads to chondrocyte dropout, which is marked by loss of cartilage and extensive subchondral bone changes. Since we all know the collagen type 2 is present in the cartilage, which is somehow degraded in the presence of some chemicals which are secreted by chondrocytes. So, chondrocyte produces matrix metalloproteinase MMPs, which is responsible for the degradation of collagen type 2, and later it is responsible for degradation of entire cartilage. So when full thickness portion of cartilage is slowed, the discharged pieces of cartilage and subchondral bone falls in joint and forms joint mice. Although chondrocytes continuously synthesize and secrete proteoglycans during disease progression, degradation ultimately exceeds synthesis and the composition of proteoglycan changes. So in response of traumatic injury, there is inflammation of the synovial joint, which is also known as synovitis, which involves infiltration of mononuclear cell such as lymphocytes, macrophage, and neutrophil into the synovial membrane, and which is responsible for the production of pro-inflammatory mediators including IL-1 beta, TNF, and chemokines in early stage and late stage of the disease. Also, TGF beta, which induces MMPs, and IL-6 can also be detected. Now, increased concentration of these mediators stimulate synoviocytes for the production of proteinase. And this proteinase, along with MMPs, is responsible for degradation of cartilage. Now there is osteophytes, which is a mushroom-shaped bony outgrowth, which also grows on margin of articular surface. And the subchondral sclerosis, which is thickening of the subchondral bone, can also be seen. Now, after the degeneration of the cartilage, subchondral bone becomes the new articulation surface. Friction with the opposite bone surface smooth and burnishes it, and gives it a polished ivory type appearance, which is known as bone hibernation. So you can see in this figure the point 1 indicates the hibernated articular surface, second one indicates the subchondral cyst, and third one indicates the residual articular cartilage. Since the two bones are in contact with each other, there are chances of getting fractures. So there are some small fractures in the bone, in the subchondral bone, which is later on filled up with the synovial fluid and responsible for the formation of fibrous wall cyst. Thanks for watching the video. If you like my video and if it was helpful to you, please like it, share it with your friends and subscribe to my channel Cyber Slate. Thank you.